So normally this lab is actually kind of hilarious because you actually keep peeing every half hour and everybody's walking through the halls carrying big beakers full of urine. And this is where people finally start like getting out of whatever. In the beginning, people start covering up their urine samples with paper towels. And it's like, I don't want anyone to see my pee or this or that. And by the end of the hour, it's just like, whatever. Um, so we're not gonna have that experience, but we will at least talk about the take-home messages and have you think about what your kidneys are doing here. And this lab is really thinking about kidney function in this kind of big picture. Like when you are coming up with your hypotheses, don't get too pedantic about things. It's more about what are the kidneys supposed to do? Water balance. If too much water is coming in, they're going to have to release more water. You're going to have more larger volume of urine. If you don't have as much water coming in, then to maintain your water balance, you're going to have to conserve your water. You're not going to pee as much. If you are taking in more salt, if you want to maintain salt balance, I didn't go into salt balance, but it's the same basic idea. Your kidneys are at the front line of your salt balance. You have aldosterone. You can tell your kidneys, keep more salt in the body or pee it out of my body. And how much you keep in or pee out depends on what the intake is, right? If I'm eating lots and lots of salty things, I need to pee out more salt in order to keep my salt balance. If I'm not, if I'm just eating like, carrots is my only meal, I'm probably going to be holding on to my salt and not peeing it out, right? So when you're coming up with your hypotheses for this lab, you really want to keep this big picture of what is the kidney doing with salt balance, with water balance. That's going to be the key to kind of doing this whole thing. So here we have this kind of doing the lab. It's just the module there that you've all got. I just added this in, so it should be good here. Just the actual PDF of the lab. So the basic idea is there's going to be 4 groups. Group 1, a group of 2, a group of 3, and a group of 4. And let me give myself my little annotator so I can like draw on this. Group 1, you're drinking 800 mils of water. That's a big gulp. That's a really large cup of water. And no salt, no caffeine. Group 2, also drinking a bunch of water, but this time you're also taking some salt tablets. So you've got, but again, no caffeine. Group 3 is getting the same amount of salt, but is only getting 1/10 of amount of water. This is like a tiny little, it's like a little Dixie cup, a shot glass size. This is basically enough water to swallow your salt tablets, but not much more. Group 4 is drinking 800 mils of water like group 1. However, they are taking 200 mgs of caffeine. That's like a very strong couple of cups. It's like a strong couple of cups of coffee. It's basically a no-dose or a Vivarin. I think we used a Vivarin in the lab. So these are the 4 groups. And normally we have people sign up into the different groups, and basically you void your bladder totally, get rid of everything that you've currently got in your bladder. And then people come in and they drink their water, take their salt or their caffeine or not. And then we start the timer. So that's like T equals 0. At T equals 0, people void their bladder. So, whatever their kidneys have been doing in the past, it doesn't matter because their bladder is now empty. And then they take their water and whatever else. And then we collect the urine every 30 minutes. So we get a sense of what is the kidney doing, because we can see what is it holding on to or what is it letting go of. We can see how much water, so in fact, let's talk about I'm sure you've all been reading the lab at this point. So what are our independent variables here? Just from what I've been talking about, what would be the independent variables?
The amount of water,
Yeah,
the
exactly.
amount of NaCl and caffeine.
Amount of H2O ingested, amount of salt ingested, the amount of caffeine ingested. So there's our independent variables. Excellent. What about our dependent variables? What are the things we're going to be looking for in response to these? Have people looked at the lab yet? People not looked at the lab?
So would they be of the urine excreted, volume, specific gravity, and sodium chloride concentration?
Exactly. We're going to look at the volume of the urine that is being excreted from the body, the sodium chloride concentration of the urine, and also specific gravity. And what does specific gravity give you a sense of? We talked about that.
Solutes.
Yeah, it's about how many solutes are in there. So we're probably going to find that NaCl concentration and specific gravity kind of move together. Typically, the more salt you have dissolved in your urine, the higher the specific gravity, the less salt, the lower the specific gravity. Specific gravity is everything, but they're going to probably be pretty related. So here we have our setup. We are changing more or less water coming in, more or less salt coming in, presence or absence of caffeine. Then we're going to see how does putting this into your body affect the volume of your urine, the salt concentration of your urine, and the specific gravity of your urine. So when you are trying to come up with a hypothesis here, what are you going to do? How are you going to think about it?
There should be 3.
I should actually, yeah, so for each of the 4 groups, you should be able to think about each of these 3 things. So there should actually be kind of 4 parts to your 12 parts to your hypothesis, right? You've got groups 1, 2, 3, 4. And for each of those, you're going to have volume, sodium chloride concentration, and specific gravity. So you kind of want to think of all of these. And how are you going to approach it? How are you going to even start thinking about what the volume is? What's the volume going to be like, or the sodium chloride or whatever? I was just talking about it just a second ago. right? What are your kidneys doing as you put water into your body or salt into your body?
Balancing it.
Exactly. So at the core of coming up with your hypotheses, you need to think about what are the kidneys going to do to restore equilibrium, homeostasis. Think about what's coming into the body and what has to leave the body to maintain a balance, right? It's all about this balancing. It's all about you want to keep your water at the, set point. You want to keep your salt at the correct level. And what your kidneys need to do in order to achieve that depends totally on what you're bringing into your body. Do I need to hold on to stuff or do I need to let go of stuff to maintain the balances? So does that make sense? Yes? No?
Yeah.
Another thing when you're making your hypotheses, there is no way you're going to be able to come up with, I think that the volume is going to be 300 mils or this or that. Everything is going to have to be relative. You can just talk about volumes, in these 4 different groups, but how are they kind of relative to each other? You can say, I think this group is going to be similar to these other groups or higher or lower than these other groups because. Same thing with sodium chloride concentration. So when you do your hypotheses, it's always going to be kind of relative because we don't have enough data to actually make quantitative predictions about what we expect in terms of actual concentration or actual volumes. We could just say, and if you use, I know like in the past years, people will say higher, like volume will be higher in group 1, but you need higher than what? You can't, higher is a relative term. Higher does not mean, you need to say higher than, you can't just say like, oh, he's taller. Taller than who? Taller than what? Taller than a gnome, he could still be really short, or like taller than Kareem Abdul-Jabbar. Well, he's like freaking huge, right? You have to say higher than or lower than. So make sure if you're using these relative terms, you say relative to what? So basically what I am going to ask you guys to do now is in your 4-person, 5-person little groups, you need to come up with your objective, which is always going to just be how are the independent variables related to the dependent variables. So that should be pretty straightforward. And then come up with your hypotheses. And again, this is going to take a little while and a little thinking because you have 4 different groups. For each group, you need to talk about the volume, the NaCl, and the specific gravity. And you have to kind of do it with relative to each other, how the groups are related to each other. Because you can't just say, oh, high or low. It has to be higher or lower than what?
Okay.
So is that making sense?
Yeah.
And then the one last, so then, what time is it? It's 11:16. So how about, I'm going to give you 40 minutes to do that. Before I do this, I want to show you the video. One very quick little video. Or actually 2 quick little videos, because this will show you more about what's going on here as well. But what? Doobie doobie doo. Doobie doobie doo. Here we go. So here is the overview.
We're going to be covering the kidney regulation of osmolarity lab today. And in this lab, we have students sign up for one of 4 groups. Group 1 is to consume 800 milligrams of water and nothing else. I'm sorry, milliliters, thank you, milliliters of water. And this cup with this red line is 800 milliliters of water. And so we want those students in group 1 to drink that as quickly as they can, as the start of the experiment. Group 2 is going to also drink 800 milliliters of water and consume 1.5 grams of salt. And here we have salt tablets. Each tablet is 1 gram, so those students would take 1.5 tablets, and they should swallow them and not chew them. And they have plenty.
So that's really, if you get lots of salt immediately into your stomach all at once to get a super hyper osmotic solution in your stomach, what's going to happen? What's that?
You'll retain more water.
Well, retain, what do you mean by retain more water?
The diarrhea.
So imagine you have super salty water in your stomach all of a sudden. What's going to happen? What happens? You have a super salty area. What happens?
Water flows in.
Water flows in. So all of a sudden you got all this water coming into your stomach, which means your stomach is starting to totally expand. And what's going to happen when that happens? If your stomach starts all of a sudden expanding and stretching?
Is it going to trigger the digestive system to move the load through faster?
Which direction?
Out. Diarrhea.
No, that's a huge long. If you all of a sudden have your stomach starting to blow up, it's just going to come out. You're going to just power hurl. So I can say, like, when I first was, this was like over 20 years ago when I was first trying to develop the lab and looking at different protocols and didn't have salt tablets. And it was like, oh, I'll just like gel cap some salt and try out, experiment and see how this works. And it was dramatic. It was like, as soon as the gel caps dissolved, all of a sudden I could just feel saliva instantly up. My mouth starts filling with saliva, and a few seconds later, I was just spewing everywhere. So, that is why you do not want to chew these salt tablets. When you take these salt tablets, they dissolve, kind of slowly and allow your body to absorb the salt and equilibrate rather than have this really that's why if you have salt water like the ocean, you can't, if you're like in a little lifeboat and you're surrounded by water, you can't just drink the ocean water to stay alive, because what happens if you drink ocean water?
That.
Exactly. Same thing, that you'll throw up. So just kind of putting that out there. Okay, let's continue now.
Plenty of water to swallow them. Group 3 is going to ingest 80 milliliters, and the red line shows 80 milliliters of water, and the same amount of salt, 1.5 grams, so 1.5 tablets of swallowing the tablets, not chewing the tablets. And in group 4, they're going to consume 800 milliliters of water and 1 caffeine tablet, which is 200 milligrams of caffeine. We're plugging, vibrating.
Good.
So each group should ingest that as fast as possible, as comfortable as possible. And then that'll be time 0. And then after 30 minutes, students will empty their bladder in various containers in using the restroom. And then they are going to take various analysis on their samples. They're going to measure their volume, and we have different sizes of graduated cylinders to hopefully match closely what the overall volume of urine that was collected every 30 minutes. Specific gravity, we'll show that in just a second. And also sodium chloride concentration, we'll show that next.
Okay, so that kind of gives you this background of what this is about. And now what I'm going to do is show you how you actually calculate or measure these things. Because you're not, if you would have been in the lab, this is what you'd be doing. So again, this is just, it's going to take a few minutes to show you how we assay the sample, and then we'll do our breakout rooms. All right, so I've been in group 3, which was just drinking 80 mils of water and taking 1.5 grams of salt. And this is a half hour after, since I did my initial voiding of my bladder and taking my treatment. And here's the amount of urine that I've got. Again, I'm collecting it every last drop because I care about the volume. So this is not a clean catch like you were doing in the urinalysis lab. You're going to collect from the beginning to the end of the flow. And first thing I want to do is figure out the volume. And I can look here roughly, it's less than 100. So when I think about which one of the cylinders makes sense, I'm going to probably take the one that's 100. This is going to get lost if I put it in one of these bigger ones here. So I'm going to pour it in. And I can go, and I can see it's about 31, 2, 3, 4, 5, 36 mils. And that is the first part. I'm also going to want to measure its specific gravity. For the specific gravity, I'm going to use this urinometer. It's basically a little vial and this float, kind of a hydrometer, like if you were making beer. It's the same idea. And depending on the density of the urine, this little float will float further up or sink further down, and the level of the urine on this little scale here is going to tell us the specific gravity. So typically you add about maybe 2/3 of the height here of the tube. If you did not pee at all, you're not going to be able to get a value. And again, if you didn't get enough urine to actually float the float, you just have no reading for specific gravity. It wouldn't be 0. 0 is not a valid, it would just be null. You don't have a value, but I have enough urine. I'm going to now put my float in, make sure it's not going to overflow. It looks good. I want to give it a little spin. I'm going to put it down here. Just kind of little style. You give it a little spin as you drop it so it doesn't stick to the walls. And then as it settles in, I can look here and it's about 1. Turn it a little. So about 0.028 is my, or 1.028 is my specific gravity for this. I'm going to mark that down. Then the last part of this is I'm going to get the sodium chloride content of my urine. For that, I am going to need a little test tube and a dropper right here. And so basically, I'm going to need 10 drops of my urine. And it matters that the drops are actual, like, well-calibrated drops. This is a quantitative test, and it depends on numbers of drops that you're adding of different reagents. So it matters that I'm careful to add exactly 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 drops of my urine. Then I'm going to add exactly 1 drop of this potassium chromate solution. And now I'm going to add silver nitrate solution drop by drop by drop. And this forms a precipitate. And you'll see as a drop hits into the solution, it's going to turn brown for a moment, but then will probably become yellow again. And I'll keep adding drops of silver nitrate until this solution here stays brown. And the total number of drops that I add here are going to be the value of the total milliliters per, or milligrams per milliliter of sodium chloride that I have in there. Again, you can see it's yellow when I start. Does this help?
Yes.
Okay, I'm going to add in a drop. Do you see little browns show up? And then 2, 3, 4, 5, 6. I need to kind of look at it. Still looking 7, 8. Oop. Not still, it's not staying brown. 9, 10. Not quite yet. 11, 12. Ooh, it's almost there. 13. I'd probably give it one more. 14. That's brown. So 14. It took 14 drops. So that would mean I've got about 14 grams of sodium chloride per liter, or 14 milligrams of sodium chloride per liter in my urine, which probably makes sense given that I'm probably pretty dehydrated being in group 3. Okie dokie. Stop sharing there. Do-do-do-be-do. So there you got it. That is what you would have been doing. And again, that is very different from the clean catch. Each time you're catching the urine at every half hour, you are just getting every last drop so you can see the total volume of urine produced. So you'll get the volume, you'll get to see the sodium chloride concentration using that colorimetric assay, and you get to get the specific gravity using the little urinometer. Although it is important, you know, when we look at the data later, especially in group 3, where they start having very little P towards the end, sometimes there is no values for specific gravity. And that's because you just can't measure it. If you can't float the float, you can't get a value. And again, no value is different from 0. No value means we don't know what the number is. So that's why when we look at the data later, you're going to see if there was not enough of a volume of urine, you were not able to actually measure specific gravity. Okay, now we're good to go. So 50 minutes, it's 40 minutes. It's 11:31. I actually have the breakout rooms ready to go, good to go. Are there any questions before I kick you all into those? So for the next 40 minutes, within your lab partners, I want you to just write down in your lab book the objective and then hypotheses. And again, hypothesis should be for each group, group 1, 2, 3, and 4, what you expect to find for the dependent variables. And again, you'll have to do it in kind of a relative way between the groups because we can't predict actual values. All right, go. All righty then. How'd it go?
Good.
So does anybody want to share Share, how about share a hypothesis for group 1?
Well, we said that if an individual consumes 800 milliliters of water with no intake of sodium or caffeine, then the subject will have the highest volume of urine, lowest specific gravity, and lowest sodium concentration.
So that's a
Compared-
good, so describing, and then you need, always remember that you always need to have the why in a hypothesis.
Yeah.
So what is the reasoning behind that? So it's a good start. Does anybody else want to kind of elaborate a little more why that would make sense?
Well, no sodium is being pumped back or pumped out of the tubule.
And why?
Because you have, because isn't it like the descending limb is not permeable to solute?
So you're getting too pedantic. You want to, again, keep it in these broader arguments around the kidneys doing this balancing. So
Could it be there's just not salt coming in, so the body, there's no reason for the body to get it out? It would reserve it for the time being?
yeah, exactly. So one thing is, if you're not taking in extra salt, especially compared, if we're doing this relative, compared to the groups that are taking in extra salt, you're going to expect the groups that take in less salt, they're going to need to let go of less salt to keep a balance. The groups that take in more salt are going to have to let go of more salt to keep a balance. Right? Does that make sense?
Yeah.
What about the volume, the water part?
Volume should be the highest.
And why?
There's nothing opposing it, like salt or caffeine.
I don't know what that means.
Okay, so if, you're not, it's just the water, there's no end, salt coming in or caffeine, it's not going to change.
Well, isn't caffeine a
coming
diuretic?
out.
So let's get back to the balancing thing. If you're taking in a bunch of water, what is your body, your kidney going to need to do to maintain balance?
Let some water out.
It's going to have to let go. So at least compared to group 3, we can say group 1 is probably going to have a lot more volume than group 3, minimally. Right? I really want you to think about the kidneys. The reason why this lab is called kidney balance of osmolarity is making you really think about the kidneys preserving this balance of water and solutes in your body. So if you're taking in a bunch of extra water, you're going to let go of more water. So at least comparing 1 to 3, that should feel pretty intuitive. And then we get into how about between 1 and 4? That's going to really depend on, depends on what your beliefs are. Does anybody have part of their hypothesis compare 1 to 4?
For each of the 4 parts of the hypothesis, are we supposed to compare each group to to all the other groups?
I'm assuming somewhere in your hypotheses, in your hypothesis for 4, do you compare it to what you found in 1?
Well, yeah, how it just has the more like it has caffeine. Group or one does not have caffeine.
Right, but in terms of volume, do you talk about comparing the volumes you expect? Like what was your Anya, what was your hypothesis for 4?
I expected the volume to be higher in group 4 than group 1 because caffeine you have caffeine added, and caffeine it makes you pee.
Right, so you did specifically compare the volume in 4 to 1 there.
But do we
Okay.
need to also, would I need to compare like 4 to like also group 2 and 3 or?
You should definitely, if it's not clear from the other relationships specified,
Mhm.
it depends on how the rest of the hypotheses are stated. There's no like kind of perfect way to do it. But so for her thing where she said 4 would have higher volume than 1, that's assuming, again, we put that thing with caffeine specifically in there because there's this kind of pop culture thing that caffeine's a diuretic. Caffeine makes you pee. So what we could do is actually check it out and see, does it seem to be supported by actual data? So we're going to when we look at the data in a few moments, we can actually check that out. It's not necessarily true or not. What about group 3 is an easy one. Who has a hypothesis for group 3?
We wrote that for group 3, it would have the lowest volume of urine, the highest NaCl concentration, and the highest specific gravity.
Okay, so again, that's a good start. And then you always want to have the reasons. Why does that make sense?
Because if you're consuming the salt and less water, your body's going to want to conserve the water, so it'll keep the salt in.
Not necessarily keep the salt. No, I mean, you're going to conserve
yeah.
the water to keep the water in because you're not drinking any water.
Yeah.
And then the salt is an independent thing here. So what's the salt side going to look like?
That one's going to be highest because it's going to release. The body has too much salt, so to balance, it's going to release some.
Exactly. I mean, and then the other thing that's going on that we're actually going to see in the actual data is, remember, concentration is amount per volume. So if you have a very low volume of urine coming out, no matter what you got in it, it's probably going to be high concentration because it's dissolved in such a small volume.
Right.
So if you have a very small volume of urine that you're peeing out, it's probably going to be very concentrated no matter what, because it's a very little volume to dissolve the stuff that you're trying to get out of your body.
I have a question. What would caffeine do to specific gravity?
It depends. It depends. I mean, you could think of it, if it really is increasing volume and nothing else, what would it do to increase? If it was just making lots more water leave your body, what would that do to specific
Would make
gravity?
it lower.
Make it lower. If it's a solute that actually needs to get excreted, if we take drugs that we put in our body, they actually leave our body through our urinary system. So we could imagine it actually increasing to some degree specific gravity by adding more solute that you're trying to get out of your body. I could see a few different arguments for it making it go up, down, or nothing. So again, it's another one where there's no correct answer. There's a few, as long as you have a prediction and a reasonable reason why that makes sense. That's all that's important. Group two. What about group 2? Ones that took a lot of water, but also took the salt.
We had, we believe total amount of water voided will be more than in group 1 because more water will be needed to release the excess salt taken in. And that the sodium chloride content and the specific gravity will increase over time and be higher than in group 1 because group 1 was not taking in any sodium.
Okay.
All right.
So if when you have that much volume already, you probably don't need, given the amount of the amount of salt in those tablets is like less than a big grab of pretzels. We actually used to use much higher levels of salt and people would start feeling off and stuff, and it ended up kind of keep revising the lab. So the amount of salt you're trying to get rid of is not that crazy. So it doesn't actually going to require more volume. You can dissolve that much salt and just add it into the urine you're doing. But it's going to make sense that you're going to have to get rid of water as well as get rid of salt because you're taking in excess water, excess salt. You're going to want to get rid of excess water and excess salt. Exactly what that's going to look like, it might be like Stephen was saying, or it might be you want to maybe balance it. You don't want to, if you get rid of too much water, then it's going to leave too much. Get rid of the water and the salt, So we're gonna, given the time, we're gonna go into the data. But in your hypotheses, if you do not have a strong explanation of why it makes sense, once we see the data, it doesn't matter if your prediction is right or wrong. Hypotheses are educated guesses. They're not supposed to be right, but they do have to have the kind of rationale. So if you don't have that explicitly in your hypothesis, after we're done with all this, if you could go back and make sure that that is kind of integrated into that, would be good. All right, so here we go. Let's see what happened. And this is actually, I took data from classes over the last few years and just put it in. It's actually kind of nice because rather than just 4 people for group, we actually get a bunch of people for group, and it's actually a little more well-behaved. Wow. All right. So let us go into the module. Here is Kidney Lab COVID edition. It should be live now. It is allowing me to load it. Oh, I got to get this into a what do you call it? Spreadsheet. Where is it? There it is. All right, so yeah, remember when you download spreadsheets, if it's in this browser, that's not going to show you everything you need. You need to open it up, download it, and open it up in the Excel proper. So let me share the Excel actual. So there it is. We've got all sorts of data, group 1, group 2, group 3. Like I said, in group 3, by the time you start getting to where they're only peeing out 8 mils of pee, there's not enough urine left to float the little float. So there's no values for specific gravity. That's why when these are blank here, that just means there was not enough urine to actually float the float. Right? You wouldn't want to put 0. 0 is not specific gravity. 0 would mean you're peeing out a vacuum or something. So let's take a look. Let's start with volumes. Let me zoom in here. I can do view, zoom. About 150 is probably right. Pretty nice. So first thing, group 3 obviously is way different than groups 1 and 2 and 4. So real obvious, all the groups that drank 800 mils of water peed out way more than the group that only drank a tiny little bit, the little 80 mils of water, the little shot glass of water. So here, just stepping back, looking at the big picture, people who drank a lot of water had a higher volume of urine coming out to keep their water balance. People who took in a very low amount of water had a very low volume peeing out because they're keeping the water in their body. They don't want to pee it out, right? So does that piece make sense? So just from the big picture, you can just think about it like that. And then we can start kind of getting more fine-tuned and look at the differences. Group 1 versus group 4, that's the water or the water and caffeine. Does there seem to be a difference? You know, maybe, sort of, kind of. If anything, if caffeine's a diuretic, it's not a very strong diuretic. You know, they might have peed a little more volume a little later, but this idea that, you know, people talk about caffeine, you know, like, you know, you're not going to stay hydrated on coffee because you're going to be peeing it all out. It's like, that's ridiculous. You can stay hydrated on caffeine. I'm living proof. You
Is that
can, sorry, go on.
Yeah, is it because of when you drink water, drink coffee, you also have water in the coffee? So
Yeah, exactly. And you're drinking a lot of water, and caffeine's not a very strong diuretic at all if it is a diuretic. So it's not like it's causing you to lose a lot of water. Alcohol is a different thing. If you're drinking a lot of beer, you might actually end up peeing out a lot more than you imagine because of it really is antidiuretic. Other thing here is we look at the group 2, and they also are peeing out a lot of water, but kind of a little bit of a less volume at any particular time. I think this is probably because they've got the extra salt, so they're getting rid of water and salt at the same time, but they're not just, when there's no salt at all, the body says, oh, there's a lot of water, let's get rid of a lot of water. But when you have a higher osmolarity in your body because you've taken the salt tablet, you're not going to be spilling out water as fast to maintain your water balance, or your osmolarity, I should say, in your blood. So I think this makes sense to me. Caffeine, if you go into the literature, the thing that actually is supported by data is it increases your intrarectal pressure. So the idea that you drink coffee and you need to poop, that is definitely something that's real. So drinking coffee and having to poop, definitely in the literature. Drinking coffee and needing to pee is not quite as supported. So it's kind of interesting there. If we look at sodium chloride concentration, again, in the big picture is kind of what you're going to expect. Group 3, the one that had very little water and ate the salt, ended up having the highest concentration. You know, we're going to look at this in more detail in a few moments. How much of this has to do with the amount of salt, and how much of it actually has to do with the fact that there's so little volume, whatever you dissolve in there is going to still be a high concentration. You know, if we look at the other groups, the concentration is all much lower. You know, groups 1 and 4. Actually, you know, group 4 has a bit of a higher concentration. Who knows why? Group 2 is higher. So this makes sense in some ways. Group 2 has a higher concentration of salt than the group that had no salt at all. And how much that could be just because there's less volume, which creates a higher concentration, or maybe there's more salt because you're getting rid of salt. We will figure that out actually in an easy way by doing, if we divide concentration, I mean, multiply. If we take salt in milliliters, oh, I got to put the units on here. Where are the units? Milligrams per mil times the number of mils you peed. That's how many actual milligrams, the actual weight, how much salt was actually leaving the body. So we can do that here, salt amount. This is just the volume times the concentration. So it's the actual, if you dried out the pee in your beaker, how much salt is sitting there in the beaker left over. And what we see is it's a little non-intuitive at first, that you actually ending up getting the least amount of salt leaving the body in that group 3. But that is because there was the least amount, there was so little urine that you had to dissolve as much as you can in it, but there's only so much you can dissolve in such a dinky amount of urine. You know, the people in the other groups, they're filling up their beakers, sloshing around. The group 3, sometimes by the end, they're not even peeing at all. So you have to have urine volume to actually carry stuff out of your body. In terms of group 1 versus group 2, we actually do see that group 2 was getting rid of more salt than group 1, which makes sense. You know, group 2 is the one that ate the salt along with the water. So they were getting rid of more salt than group 1 was because they had more salt they needed to get rid of to keep their balance. This is something we do notice pretty regularly, that whatever the caffeine is doing, it seems to increase the salt over the baseline. So this is not just some spurious result. It seems to happen pretty regularly. I'm not sure why. I have no idea why. So if we look at, I have to get rid of my dealy bow. So specific gravity is basically going to mimic the sodium chloride concentration, right? If we look at the sodium chloride concentration, very high concentration of salt in group 3, low in the other groups. Specific gravity, it's the same basic deal. So that should not be surprising. So again, take home messages. I really want you to just think about your kidneys helping maintain homeostasis, this osmolarity, the amount of salt versus the amount of water in your body. You know, yes, they do help you get rid of waste. That is an important role they play as well. But, you know, they are critical just moment by moment, making sure that your osmolarity stays right and that your water balance is right and your salt balance is right. So in your discussion, you know, do your best to address your hypotheses in relationship to these results. Do these results support your hypotheses or not? And then in the bigger picture, also just kind of talk about the basic story that these results tell us about, you know, what the kidneys are doing in terms of water and salt balance and how that is important for maintaining our basic homeostasis. So are there any questions about all this? Okay, so what I'm going to do sometime in the next, either by tonight or tomorrow, I will put a submission thing for this lab in as well. It'll be a part of the module. But after this, there's only one more lab, which is kind of amazing. We're going to do the digestion lab, which doesn't make sense to talk about yet until we start introducing digestion, which I will talk about next week. And then the only other things left in lab are going to be kind of reviewing for the lab exam, taking the lab exam, and doing our presentations.